Hello dear children, welcome back. I'm Dr. Jisha Joe. In this session, we shall study about the endomembrane system. Now we know that cell has different cell organelles. What are these cell organelles actually? They are living parts inside the cell with specific structure and specific function. In the previous videos, we have studied about the structure of chloroplast, mitochondria. These are cell organelles. They have a specific structure and specific function. But most of the cell organelles work in a coordinated manner. For example, as in the case of endoplasmic reticulum, the Golgi Apparatus, the lysosomes, vacuoles, etc. If you look at all these cell organelles, we can see that these cell organelles function together or work together in a coordinated manner. And these together constitute what we call as the endomembrane system. Okay, so the cell has several cell organelles. The cell organelle, each cell organelle is a living part of the cell which has specific structure and specific function. But most of the cell organelles function together or work together in a coordinated manner. And these together constitute what we call as the endomembrane system. So as I told before, the endomembrane system consists of the endoplasmic reticulum, the Golgi apparatus, the lysosomes and the vacuoles. So we shall study each of these in detail. First, let us start with endoplasmic reticulum. The term endoplasmic reticulum was coined by Porter. Porter coined the term endoplasmic reticulum, and the endoplasmic reticulum consists of a network of paired membranes scattered in the cytoplasm. Okay, network of paired membranes scattered in cytoplasm. This endoplasmic reticulum divides the intracellular space. What is the intracellular space? That is the space inside the cell. Okay, intracellular space is divided into two compartments by the endoplasmic reticulum. So if that is the cell that is the cell membrane. Okay, we have the nucleus. Okay, double-layered nuclear membrane. We will study about the nucleus, the structure and function later. So the endoplasmic reticulum can be seen arising from the nuclear membrane. You can see okay. So here there are several endoplasmic reticulum arising like that. This is just two I have drawn here, okay. This is just two. Okay, so you can see that is the endoplasmic reticulum. As you can see, this is the intracellular space, that is the space inside the cell. The endoplasmic reticulum divides the intracellular space into two compartments. Which are the two compartments? That is the space inside the endoplasmic reticulum, which is called as the luminal compartment. Luminal compartment. And the space outside, that is the space, which is the extraluminal compartment. Extraluminal compartment. So that is the third point. The endoplasmic reticulum divides the intracellular space into two compartments. The luminal compartment, that is the space inside the endoplasmic reticulum, and the extraluminal compartment, which is the space outside the endoplasmic reticulum. So I hope that much is clear. So endoplasmic reticulum, the term endoplasmic reticulum was coined by Porter. Endoplasmic reticulum consists of a network of paired membranes scattered in the cytoplasm, as you can see there. And the endoplasmic reticulum divides the intracellular space into two compartments, the luminal compartment and the extraluminal compartment. Okay, now from where does it originate? As you can see, the endoplasmic reticulum originates from the nuclear membrane. Okay, origin from nuclear membrane. The endoplasmic reticulum originates from the nuclear membrane, and it is absent in prokaryotes. Absent in prokaryotes, but present in all eukaryotic cells. Present in all eukaryotic cells except RBC. So, mature RBCs lack endoplasmic reticulum. So, endoplasmic reticulum is absent in all prokaryotic cells, and also it is absent in RBCs, whereas endoplasmic reticulum is present in all eukaryotic cells. Mature RBCs lack endoplasmic reticulum. The RBCs have to travel through the minutest capillaries to reach the cells. So, if endoplasmic reticulum was there, endoplasmic reticulum provides mechanical support to the cell. It gives a framework for the cell. It acts as a mechanical support to the cell. So if it was present, RBCs would not be flexible enough to move through the capillaries. That is the reason why RBCs do not contain endoplasmic reticulum. So, endoplasmic reticulum is absent in prokaryotic cells. It is present in all eukaryotic cells except mature mammalian RBCs. Okay, so I hope that much is clear. So, endoplasmic reticulum, the term was coined by Porter. It consists of network of paired membranes scattered in the cytoplasm. Endoplasmic reticulum divides the intracellular space into two compartments, the luminal compartment and the extraluminal compartment. It originates from the nuclear membrane. It is absent in all prokaryotic cells and in mature mammalian RBCs. It is present in all eukaryotic cells. Endoplasmic reticulum exists in three forms. Okay, in three forms, or there are three components in the endoplasmic reticulum. They are the cisternae, the vesicles, and the tubules. These are the three components of endoplasmic reticulum, that is cisternae, vesicles, and tubules. Now, what are cisternae? Cisternae consists of unbranched flat sheets lying parallel to each other. You can see flat sheets which are unbranched lying parallel to each other. These are called as the cisternae. So, endoplasmic reticulum can exist in this form, as unbranched flat sheets lying parallel to each other, and these cisternae bear ribosomes. Ribosomes might be present attached to the cisternae. You can see ribosomes attached to the cisternae. Okay. The second form in which endoplasmic reticulum can exist is the vesicle. Vesicle comprises of spherical bodies. They are spherical bodies. So, endoplasmic reticulum can exist also in this form as spherical bodies. When they exist as spherical bodies, they are called as the vesicles. And vesicles also bear ribosomes. Ribosomes are seen attached to the vesicles. And the third form in which endoplasmic reticulum exists is the tubules. Tubules are tubular branching elements. As you can see here, they are branching elements These are the tubules, and ribosomes are mostly absent in tubules. Okay, you rarely see ribosomes in the tubules. No ribosomes will be present in the tubules. So these are the three forms in which endoplasmic reticulum can exist. Okay, they are the cisternae. They are flat, unbranched sac-like structures lying parallel to one another. They bear ribosomes. Second form, vesicles. They are spherical bodies. Again, these also can bear the ribosomes. And the tubules are tubular branched elements, and they do not contain ribosomes. They normally don't bear the ribosomes. Okay, so these are the three forms in which endoplasmic reticulum can exist. And based on the presence or absence of ribosomes, endoplasmic reticulum is classified, or it is categorized into two. Okay, based on the presence or absence of the ribosomes. So if ribosomes are present, they are called as the rough endoplasmic reticulum. Rough endoplasmic reticulum, ribosomes are present. Such type of ER are called rough ER. If ribosomes are absent, then they are called as the smooth endoplasmic reticulum. So we know the function of the ribosomes you have studied in your lower classes. The function of the ribosomes is, ribosomes are the sites of protein synthesis. So wherever rough endoplasmic reticulum are present, they will be regions where proteins are synthesized, because such endoplasmic reticulum have ribosomes in them. So in protein synthesizing cells, or cells which are actively involved in the synthesis of proteins, you can see rough endoplasmic reticulum. Whereas smooth endoplasmic reticulum is present in cells concerned with lipid metabolism or glycogen metabolism. Cells that are concerned with the synthesis of lipids or the metabolism of lipids or glycogen, you can see smooth endoplasmic reticulum. In smooth endoplasmic reticulum, ribosomes are absent. Okay, so the ribosomes give mechanical support to the cell, and the rough endoplasmic reticulum are found in cells which are actively involved in protein synthesis because of the presence of ribosomes, whereas the smooth endoplasmic reticulum are present in cells concerned with lipid or glycogen metabolism. Now moving on to the Golgi apparatus. Golgi apparatus were discovered by Camillo Golgi, and that is how this organelle gets its name as Golgi apparatus. It was discovered by Camillo Golgi. It has high lipid content, and in plant cells the Golgi apparatus is called as dictyosomes, whereas in animal cells they are called as Golgi apparatus. In plant cells they are known as dictyosomes. The Golgi apparatus consists of flat disc-like sacs placed parallel to one another. As you can see here, flat disc-shaped sacs placed one above the other. Okay, so that is the Golgi apparatus. As you can see, they are flat sacs placed parallel to one another, and that is the Golgi apparatus. As we have seen the cisternae of the endoplasmic reticulum, here also the flat disc-like sacs, they are called as the cisternae, and they are placed parallel to one another. So that is how the Golgi apparatus look like. So the first point, it was discovered by Camillo Golgi. Second point, they have high lipid content. Third point, they are called as dictyosomes in plant cells, and the Golgi apparatus consists of flat disc-shaped sacs called as the cisternae that are placed parallel to each other. The Golgi apparatus, as we have seen before, they consist of flat disc-shaped structures which are called as the cisternae, arranged parallel to one another. The Golgi apparatus are seen mostly close to the close to the that is true, close to the nuclear membrane and the endoplasmic reticulum. Okay, and if suppose that is the nuclear membrane, you can see the disc-shaped Golgi apparatus like that. Okay, That is the Golgi apparatus, the cisternae of the Golgi apparatus. You can see the Golgi apparatus there. If you look at each disc shaped sac, you can see that it has a convex side and a concave side. That is the convex side and that is the concave side. The convex side is the cis face. Okay, the convex side. So I'm just enlarging one. Okay, that is one of it, one of the cisternae, that is the nuclear membrane there. Okay, so Golgi bodies are seen close to the nuclear membrane and also seen in association with the endoplasmic reticulum. And if you look at the Golgi apparatus, it has each sac-like cisternae will have a convex face, that is the convex side, which is called as the cis face, or it is called as the forming face. And a concave side, which is called as the trans face. That is the trans face or the maturing phase of the Golgi body. Maturing phase of the cisternae. So you can see the convex side. The convex side is the cis phase or the forming phase, and the concave side is the trans phase or the maturing phase. And as I told you, the convex phase will be facing, or the cis phase will be towards the nuclear membrane, towards the endoplasmic reticulum, whereas the concave phase, that is the trans phase, will be towards the cell membrane or the plasma membrane. So if I draw the cell membrane there, that is the cell membrane. Okay, that is the cell membrane. You can see the cis phase is towards the nuclear membrane or towards the endoplasmic reticulum, whereas the trans phase is seen towards or close to the plasma membrane. Okay, this is how the Golgi apparatus or the cisternae in the Golgi bodies is arranged, with the cis phase towards the nuclear membrane or in close association with the endoplasmic reticulum, and the trans phase in close association or very near to the plasma membrane. Now let us move on to the functions of the Golgi apparatus. Okay, the first function is the storage of materials. Golgi apparatus is concerned with the storage of substances. Second function, it is concerned with the formation of glycoproteins and glycolipids. So they are the important sites for the formation of glycoproteins and glycolipids inside the cell. Then third point is that the Golgi apparatus is concerned with packaging of materials. The packaging of materials to be transported within the cell or to be secreted outside is done in the Golgi apparatus. Okay, the materials that are to be transported within the cell or that are to be secreted outside the cell are packaged within the Golgi apparatus. That is, the packaging of materials to be transported takes place in the Golgi apparatus. The last point is the modification of proteins. The proteins which are formed get modified and they fuse to the forming phase. The proteins, we know the ribosomes are the sites of protein synthesis. As the proteins are synthesized in the ribosomes, they get transported or they form vesicles and they move to the cis phase or the forming phase of the Golgi apparatus, and they fuse with the cis phase or the forming phase, and they get modified and are finally released out through the trans phase. So as vesicles, We know what are vesicles. They are spherical bodies, isn't it? As we have seen in the endoplasmic reticulum. So as vesicles, they fuse with the cis phase and they get modified. Modification of the proteins take place inside the Golgi apparatus, and the modified proteins are released out through the trans phase, that is the concave phase or the concave side of the Golgi apparatus, the maturing phase, which is seen towards the cell membrane or the plasma membrane. So these are the functions of the Golgi apparatus. So first function, it is the storage of materials. Second function, it is the important site for the formation of glycoproteins and glycolipids. Third function, it is concerned with the packaging of materials to be transported within the cell or materials to be secreted out. Materials to be transported out of the cell are also packaged within the Golgi apparatus, and Golgi apparatus is concerned with the modification of proteins. So substances that are to be modified are brought as vesicles. They form vesicles, and these vesicles fuse with the cis phase, that is the forming phase, and they get modified inside the Golgi apparatus, and are after modification they are released out through the trans phase. The trans phase is seen close to the plasma membrane or the cell membrane. So I hope that is clear to you. Now moving on to the lysosomes. Lysosomes are membrane-bound vesicles that are formed by the packaging in the Golgi apparatus. Okay, they are membrane-bound vesicles that are bounded by membrane, and they are formed by the packaging in the Golgi apparatus. These lysosomes contain hydrolytic enzymes. Hydrolytic enzymes are digestive enzymes. They are also called as hydrolases. They may be lipases, protease, carbohydrates, etc., which are concerned with the digestion of lipids, proteins and fats. They are the hydrolytic enzymes. They are digestive enzymes. They are also called as hydrolases. Okay, they may be lipase, protease or carbohydrates, which are concerned with the digestion of lipids, proteins or carbohydrates. So, and these, as we have seen, lysosomes are called as the suicidal bags of the cell because they are concerned with the digestion or the destruction of the worn out cells. Since they are concerned with the destruction of its own cell or the cell in which it is present, that is why it is called as the suicidal bags of the cell. The worn out or destroyed or damaged cells are destroyed inside the lysosomes. With the help of the hydrolytic enzymes present in the lysosomes, the cells get destroyed, and that is why they are called as the suicidal bags of the cell. And as we know, these enzymes, as I told you, they are the digestive enzymes, and they are active in acidic pH. All the enzymes, hydrolases, are active in acidic pH. Lysosomes are also concerned with the destruction of foreign substances or foreign particles or germs that has entered into the cell, and also they help in the digestion of food. Okay, so that is with regard to the lysosomes. So lysosomes are membrane-bound vesicles formed by the packaging in the Golgi apparatus. They contain hydrolytic enzymes which are otherwise called as the hydrolases. They are concerned with the destruction of the damaged cells, and also they are concerned with the digestion of food, and also the destruction of any foreign particle or germ that has entered into the cell. So that is with regard to the lysosomes. Now if we look at the vacuoles are spaces inside the cytoplasm which are bounded by membrane. That is, they are membrane bound spaces, and these membranes, it is just a single membrane that bounds or surrounds the space, and this single membrane is called as the tonoplast. So the membrane bound spaces within the cytoplasm is called as vacuole, and the membrane of the vacuole is called as the tonoplast. It is just a single membrane, and the vacuoles contain the sap sap refers to the water with all the dissolved minerals as you see in plant cells. Then it may contain water, waste products, etc. will be present inside the vacuole. Then in the case of plants, if we compare the vacuoles in plant cell and animal cell, we can see that in plant cell vacuoles are very large, and whereas in animal cells vacuoles are very small or even absent in the case of animal cells, whereas in plant cells we can see very large vacuoles. And if you look at the concentration of the cell sap, the sap inside the vacuole, we can see that the concentration, or it is highly concentrated. The cell sap of the vacuoles in plant cells is highly concentrated, and this will help or facilitate the uptake of ions in plant cells. Okay. Now, in the case of amoeba or protistans, you can see a type of vacuole which is called as the contractile vacuole. These contractile vacuoles are concerned with osmoregulation. What is meant by osmoregulation? Osmoregulation is the maintenance of the osmotic pressure inside the cell. So, the contractile vacuoles present in the amoeba and other protists help in osmoregulation, or helps in maintaining the osmotic pressure of the cell. So that is with regard to vacuoles. So vacuoles are membrane-bound spaces within the cell. The membrane of the vacuole is called tonoplast. Vacuoles contain sap, water or waste materials. In plant cells, the vacuoles are large, and the concentration of the cell sap in the vacuoles is very high, and this facilitates the transport of ions against the concentration gradient in the case of plant cells. When we move on to animal cells have very small vacuoles, or sometimes even absent. Vacuoles are absent in animal cells. When we move on to amoeba and other protistans, you can see that contractile vacuoles are present, which help in regulating or maintaining the osmotic pressure of the cell. So dear children, in this session we have studied about the endomembrane system, which consists of the endoplasmic reticulum, the Golgi apparatus, the lysosomes and the vacuoles. I hope this session is clear to you. If you have any doubts or suggestions, please ask your doubts and give your suggestions in the comment section. Thank you.